Drop a single chemical into a clear fluid, and the solution might flash vibrant pink, then immediately shift to deep blue. These intense reactions are the signature of transition metals. We see this image of microscopic red blood cells, which exist entirely because transition metals act as the chemical engines of human biology. Iron sits at the core of hemoglobin, binding and releasing the oxygen, keeping you alive. This happens through a specific structure called a coordination compound. A positively charged transition metal acts as a Lewis acid at the center. It pulls in surrounding molecules or ions called ligands, which act as Lewis bases by donating their electron pairs to the metal. The exact sequence and orientation of how these ligands lock into the metal determine the physical behavior of the entire compound, from the toxicity of a heavy metal to the deep hue of an industrial dye. Mapping those rules began in the 1890s with chemist Alfred Werner. He proved that coordination complexes do not form at random. Instead, they organize into fixed, predictable three-dimensional geometries based on primary and secondary valencies. Werner's work revealed the existence of structural isomers. You can take the exact same atomic ingredients and connect them to the central metal in completely different orders. Ions can swap positions inside and outside the coordination sphere, creating entirely distinct molecules with different properties. Then there are stereoisomers. Here, the exact same atoms share the exact same connections, but they differ entirely in their 3D spatial arrangement. They can sit adjacent or opposite to one another, or even form mirror images of the molecule that cannot be superimposed on each other. Ligands can also exhibit the chelate effect. Instead of connecting at a single point, large molecules with multiple donor atoms bite the central metal multiple times, wrapping around it to form highly stable, interlocking ring structures. We can look directly at these 6 distinct samples of cobalt complex powders. Each one has a different arrangement of ligands. Werner mapped the physical geography of these compounds, but his structural rules offered no explanation for why they formed these specific shapes or where these brilliant colors actually came from. The answer arrived in the 1930s with crystal field theory. This model treated the approaching ligands simply as static negative point charges rushing toward a positively charged metal center. Here are the 5 degenerate d orbitals of a transition metal. As 6 negative ligands approach, they repel the electrons. The 2 orbitals directly on the axes are pushed higher in energy, while the 3 between the axes stay lower. The orbitals split. This side-by-side diagram compares that octahedral split to a tetrahedral field. When only 4 ligands approach between the axes, the splitting pattern inverts, and the energy gap between the upper and lower orbitals is significantly smaller. The exact 3D arrangement of the external ligands strictly dictates the internal quantum energy landscape of the metal's electrons. The energy gap dictates interaction with visible light. An electron jumps to a higher orbital by absorbing a precise photon. If it absorbs a red photon, red is subtracted from the spectrum. The remaining light passing through to your eye is the complement, green. This internal energy system also drives physical structure. According to the John Teller theorem, if electrons are unevenly distributed, the complex becomes unstable. The molecule will physically distort, elongating specific bonds to break the degeneracy and settle into a lower overall energy state. Crystal field theory elegantly links geometry to color, but it harbors a massive flaw. If ligands act exclusively as static negative magnets, then highly charged ions should cause extreme splitting. Instead, real-world data proves that neutral molecules cause the largest energy gaps. The electrostatic model is incomplete. Ligand field theory provides the ultimate solution. It moves beyond static electricity by integrating molecular orbital theory, accounting for actual covalent electron sharing between the metal and the ligands. Look at this directional flow diagram. Ligands donate electrons to the metal, but some ligands don't stop there. Through a process called pi back bonding, they actually accept electron density back from the metal, filling their own empty orbitals. This explains the anomaly with neutral molecules like carbon monoxide. It is a powerful pi acceptor. As the metal pumps electron density back into the carbon monoxide, It drastically widens the energy splitting gap in the metal's d orbitals. Coordination bonds are not static one-way magnets. They are highly dynamic two-way quantum channels of electron flow. This portrait shows chemist Henry Taube, who pioneered the study of how these quantum rules dictate real-world chemical kinetics. He mapped why some labile complexes swap their ligands in a fraction of a second, while inert complexes take days to react. As you can see in this comparison, ligands swap out through specific mechanisms. In a dissociative mechanism, the complex loses a ligand first, dropping to a 5-coordinate intermediate. In an associative mechanism, it forces a 7th ligand into the structure before kicking one out. We also see directional traffic control, like the trans effect in square planar complexes. A strong tie acceptor ligand will pull so heavily on the shared electron density that it will literally fracture the bond located exactly opposite, or trans, to itself. When metal complexes trade electrons in redox reactions, they use these same spatial rules. They either share a bridging ligand to pass the electron through an inner sphere mechanism, or they allow the electron to jump through space between two intact complexes in an outer sphere mechanism. From Alfred Werner sorting colored powders by their macroscopic geometry to the mapping of two-way quantum pi backbonding, coordination chemistry reveals how structure dictates function. Understanding the invisible architecture of d orbitals gives us the exact tools needed to manipulate the tangible molecular machinery operating in everything from industrial catalysis to human blood.